is Jakub Kopal from the University of Oslo. He's going to tell us about copy number variation in adolescence.
Good afternoon. So yes, my name is Jakub Kopal, and I am from the University of Oslo. I'm a postdoc there, and this work we've done before with Daniel Obzdok and Sébastien Jacquemont when I was doing my postdoc at McGill University in Montreal. And there we were studying the relationship between genes, brain, and behavior. It's needless to say that this relationship is really complex, and it cuts across disciplines when you try to study that. And the genetic architecture really plays a crucial role in shaping our development at all the stages of our lives. But in this particular talk, I will focus on adolescence, which is really a critical juncture in our development. That can be illustrated, for example, by the fact that mental illness typically manifests during adolescence. And so there is really a multitude of factors that shape the neurodevelopment trajectory, but currently most of the studies, they focused typically on one or few such factors at a time. So instead we thought, with the great resources that we have now available, let's really do an interdisciplinary approach that will cut across sociology, psychology, biology, and neuroscience in a single study. So that's why we use the great resource that is ABCD, which is the largest neurodevelopmental imaging resource, 10,000 kids with measures of cognition, but importantly also genetics, brain architecture, psychosocial, and socioeconomic measures. And we try to do a multidimensional understanding of the interplay between genes, brain architecture, and behavior. And when we talk about genetic architecture, we typically talk about GWAS studies, human biology studies, but there are several limitations, such as that identified SNPs tend to reside in non-coding regions, or they are associated with small effects on the phenotypes under study. So that's why we focus on copy number variations, which are deletions or duplications of large genomic segments, and they were associated with body-wide implications. So here is a figure of a famous 16p deletion duplication, and you can see the associated phenotypes and also the genes that are encompassed. So as a first step, we needed to identify CNVs in the ABCD resource. So we found out there are 5,500 controls, meaning adolescents without any large CNV. There were more than 500 adolescents that carry CNV that encompass at least one gene, and there were more than 1,400 adolescents with a duplication. There were no differences in sex or age between these three groups, and most of the CNVs encompassed a single gene. And as I said, multidimensional understanding, so we used a latent variable approach to derive population modes of covariation, trying to link brain and behavior. So on the side of the brain, we had 148 regional volumes according to the Destry Atlas. On the side of the behavior, we had almost 1,000 behavioral phenotypes that cuts across 20 categories. And we try to quantify the effect of carrying a CNV on the revealed modes of population covariation. As a tool, we used canonical partial least squares. So to give some more details, first we used PLS to project brain and behavior measurements into low-dimensional subspace where the covariation between the projections is maximal. Using several techniques, we found out there are 3 significant PLS modes, and then we re-expressed brain and behavior measurements of CNV carriers using the model that was derived using controls. Finally, in the next step, we evaluated the differences in the expression of brain and behavior modes between the controls, CNV deletions, and carriers of CNV duplications. And to introduce you to my modes of population covariation, so here is the dominant mode. I'll go slowly through that. So on the side of the brain, we can look at the loadings. Loadings tell us how each particular brain phenotype contributes to the low-dimensional projection. So on the brain side, we see high loadings in the middle occipital sulcus, lingual gyrus, cultural gyrus, cuneus, summarized by the 7 large-scale networks according to Schaefer-Yeo atlas. We see strong loadings across all 7 networks. And this particular brain mode was highly correlated or covariate significantly with a behavioral mode that can be characterized by high loadings in the fluid cognition, crystallite cognition, highest grade, family income, violence, and education. If we summarize the behavior phenotypes by categories, we see highest loadings in the socioeconomic, demographics, and cognitive domains. And finally, for the influence of a CNV on the expression of these modes, so we saw that carrying a deletion leads to a significantly different expression of the behavior mode for both deletions and duplications, and a significantly different expression of the brain mode in duplications. What this means that carrying a CNV leads to diminished cognitive functioning and also real life functioning. Now for the second mode that can stay hidden in classic analysis, mode that is orthogonal to the first mode. So in the second mode, we observed an opposite effect of deletions and duplications on the brain pattern, and we also observed a significant effect of deletions on the expression of the behavior pattern. We can again look at both of the sides. So first, on the brain pattern, on the brain side, we see a high loading in the lingual gyrus, supremolgen gyrus, precentral gyrus, regions that mostly belong to frontoparietal network, but also dorsal attentional network or somatomotor network. On the behavior side, we see strongest loadings with phenotypes that come both from the child behavioral checklist, but also from the adult self-report questionnaire. So phenotypes such as total problems, thought, hyperactivity, anxiety, depression, externalizing problems, parent behavior. So importantly, phenotypes from both the parent side, but also from the child side. So this really shows that CNVs can influence the entire family environment, the family dynamics. Finally, third significant mode. In the third mode, we observed a significant effect of deletions on the expression of the behavior mode. And this mode can be characterized by strong loadings in the in the default mode network and the frontoparietal network, so the deepest layers of the human cortex. And on the behavior side, we see strong loadings, mostly in the socioeconomic category, but also in the demographics. The leading phenotypes in this mode were crime reports, drug abuse, drug state, psychosis, or percentage of owners. So really, and also importantly, violence in the neighborhood. So more of a environmental, socioeconomic phenotypes. In the next step, we look if what we observe is actually due to the presence of a CNV, or it can be explained by some sources of population stratification. So we looked for the associations between the expression of our brain mode, of the behavior mode, with several metrics of population stratification, but we did not observe any association between sex, ethnicity, age, site, or genetic PCs with the expression of a brain mode. So that really shows us that whatever we observe is driven by the presence of the genetic mutation rather than some hidden source. So as the main conclusion, hopefully what we showed is that CNVs, they have impact that go well beyond just cognition and language that are typically reported in the literature. And the real life consequences go really far and are far-reaching, and we can observe that on the family environment, but also, for example, on the neighborhood dynamics. So with that, I would like to thank my collaborators from both of the labs. We have a preprint online, and also if you have questions, happy to answer now or at my poster on Wednesday and Thursday. Thank you very much.
Thanks, Jakub, for an excellent presentation. Are there any questions from the audience, perhaps?
Yeah, thank you for your interesting presentation. Actually, I'm lost at that how to use a single value to represent the behavior in the same place, same space with brain data. Yeah, I don't understand how to convert the behaviors into a.
How to convert behavior into a single number?
Yeah.
Well, you know, it's you can picture it as running a PCA on the behavior. So yeah, it's a low-dimensional projection. It's a particular behavior pattern, and we're just looking at the expression of the behavior pattern. The behavior pattern can be characterized by several behavioral measures that are highly correlated. So instead of looking, you know, at one behavioral phenotype at a time, we look at them in like more of a global perspective, recognizing the correlations between the different behavioral measurements.
Okay, I think I got that. Thank you.
Can I maybe ask one quick follow-up question if there's none? There's a question there if you want to maybe use the microphone.
So thank you very much for your talk. Again, something that I also didn't understand about the behavior. So the behavioral measures that you have, were they in any way grouped together through some kind of a PCA analysis or something like that? Because if you want to understand the mechanism that what drives there on the behavioral level, there can be a lot of backward causalities and things like that that will influence how different modes of the behavior are related to the genetic and to, not to the brain so much, but to the genetic. Did you try to make any kind of an advanced analysis over there of the behavior and to see what drives what in the behavior and to group them together?
So no causality, purely correlation.
Yeah, okay, the
analysis. Yeah, well, so one approach is really go phenotype by phenotype, right? Or you kind of take this bazooka approach and try to look at all the behavioral and all the phenotypic information that you have about the participants, and take it as a more of an exploratory analysis that can generate further hypotheses that can be tested. So we did not focus specifically on any phenotype or any category. We wanted to keep it as broad as possible.
Okay. We can.
But yeah, we can, I guess, talk later. I'm happy to answer.
Thank you, Jakub.